As a child, I was always fascinated by nature and by computers. I experimented with plants and animals around the house. I learned programming, and I even analyzed my experimental results on my father's PC. Deciding what to study was a tie between computer science and biology. I had to choose, so I chose for biology, but my computer fascination never faded away. After my studies, I started a PhD in the young field of proteomics, because I really wanted to understand how living systems function on a molecular level. But during that PhD, I already encountered and was happy to tackle several bioinformatics problems. In the early 2000s, life sciences underwent several disruptive changes. For the first time ever, we got full DNA sequences for organisms available. With mass spectrometers, we could analyze thousands of proteins simultaneously instead of just a few. Of course, this all brought significant computational challenges. In that period, the speed at which we could collect molecular data started to increase more rapidly than our computer power increased. So data analysis really became a bottleneck, and a lot of life science data remained hard to interpret with the existing computational tools. I then realized that biology needed a data science perspective. It was during a postdoctoral research stay in Denmark that I decided to full-time shift my attention to bioinformatics. When I came back to Antwerp, I started to work in the Department of Mathematics and Computer Sciences with data scientists. And I founded, together with colleagues of the University Hospital Biomina, the Biomedical Informatics Network Antwerp, that had as a goal to bring together life scientists and data scientists around the typical biocomputational challenges. I like to explore nature with family, and I'm also very passionate about playing music. I play violin and double bass, mostly vintage American roots music. In 2015, I started my own multidisciplinary research team. The goal of our team is to explore novel approaches to make sense of large-scale molecular data. In particular, we study frequent pattern mining methods that can reveal novel associations, novel hypotheses regarding complex molecular data. We also often study machine learning approaches that can be used to develop predictive models with regards to molecular profiles. Our approaches have already generated fundamental insights in molecular processes. For example, we have elucidated some of the rules that underlie the recognition of an intruder by the immune system. This has possible applications in vaccine development or in cancer treatment. We also apply our approaches in entirely different areas, ranging from toxicology to plant biotechnology. From a clinical perspective, your DNA and other molecular information is extremely informative and very personal. I hope that one day our algorithms can be useful to turn such information into recommendations for personalized medicine that make a difference in the clinic.